Hello everybody, and today I'm going to be talking about mitochondria, and specifically I'm going to be talking about mitochondria as potentially being the root cause of essentially all major chronic diseases. So it is my hypothesis that the cause of essentially all of the major chronic diseases is primarily foundationally related to mitochondrial function. Things like cardiovascular disease, diabetes, cancer, neurodegenerative disease, autoimmune disease. All of these diseases have been seeing increases within the last decade in the potential for mitochondrial dysfunction to be at the root cause. And unfortunately, though, I think that the treatment of these diseases, looking through a mitochondrial lens, has not caught up yet. And so that's what I want to be talking about today, is not only that the potential of these diseases to be originating from mitochondrial dysfunction, but also for these diseases to potentially be treated better when looking at the diseases through a mitochondrial lens. And so mitochondrial biology has many implications in all of the major chronic diseases that I just talked about, but it also possibly has other implications, potentially increased sports injuries, increased disc herniations, and maybe impacts basically everything that has to do with human health. Okay, so the hypothesis that I'm specifically presenting today is that essentially all of the major chronic diseases are diseases not of the genes in the nucleus, but that they are diseases of the mitochondria, that they specifically are tissue-specific, energy level-specific drops in mitochondrial energy production. And again, it's the mainstream view today is that many of these diseases are diseases of the nucleus and of genes in the nucleus that undergo mutations. But what I'm proposing today is that that is not the case for the fundamental cause of many of the chronic diseases that we face today, and that in fact the actual true root cause of these diseases is a drop in mitochondrial energy production, and that this drop is a tissue-specific, energy level-specific drop in mitochondrial energy production from dysfunction of the mitochondria. Okay, so what's the evidence for this? So this was a case study. This was a family that Dr. Doug Wallace studied in the early 1980s that had a specific mitochondrial DNA mutation. Before we go any further, so Dr. Doug Wallace is the man that found that all of the mitochondria that we inherit come from our mother. So we don't inherit any mitochondrial DNA from our father, unlike the nuclear genes where we go through the traditional mixing of the genes. So we inherit all of our mitochondrial genes from our mother. Dr. Doug Wallace found that out. He's been researching mitochondria for about 40 years, and he's done a lot of the major research in this field. So this was a case study of a family that he studied. So the mother and her kids had a specific defect in their mitochondria. They had a defect in the tRNA leucine gene at A3243G. That's not important to memorize, but you can see that the conditions that they had on the slide. So this is a representation of the family. You see the mother had these issues, and then essentially all of the kids had these issues. So they had lactic acidosis, growth retardation, early onset dementia, stroke-like episodes, hypertrophic cardiomyopathy, and conduction abnormalities. Interestingly also, so the oxidative type 1 muscle fibers were degenerated with these abnormal mitochondria. They turned into what's called ragged red fibers called mitochondrial myopathy. Well, the glycolytic fibers were perfectly fine. That makes sense because this was a mitochondrial disease. There was a drop in mitochondrial energy production. So it affected the cells that relied more on mitochondrial energy production, and as opposed to the ones that basically relied on glycolysis, which occurs in the cytoplasm mainly, or exclusively. Okay, so this is perhaps the most important slide in this presentation, and this basically gives an encapsulation of the central idea of this point. So what you can see is from that family, so all of the young men and women either died of a status epilepticus or cardiac dysrhythmia in their late teens or early 20s. So the family had about 70% mutant that gave them myopathy, cardiomyopathy, stroke-like episodes, so-called Mila syndrome. However, though, what's really interesting is that if you take that same mutation and you take it down to 20 or 30% mutant, it gives you type 1 diabetes, type 2 diabetes, or autism. And you can see that here. This was from a Dr. Doug Wallace study that they did. So the family had 70% mutant, and they had the lactic acidosis, the growth retardation, the cardiomyopathy, those things. However, if you take that same DNA mutation, but you drop the mutation rate down from 70% to 20 to 30%, you end up with type 1 diabetes, type 2 diabetes, or autism. And if you took that mutation in that same gene to 100% mutant, you get the Lee syndrome and the perinatal lethality. It's a lethal childhood disease. So what's interesting is that the exact same mutation in the exact same gene, but to different degrees, which results in a different level of drop in mitochondrial energy production, it gives you totally different diseases, different phenotypes. And so the question is why? And so what that tells you, and we're going to get into this more when I talk about mechanisms, is that there's these specific nuclear phases that respond to a continual change in mitochondrial energy production. So what's essentially happening is that the mitochondria and the mitochondrial energy production and the efficiency of mitochondrial energy production in the mitochondria is actually controlling much of the nuclear transcription, and genes are literally being turned on and off, and that's being controlled by the mitochondria and how essentially healthy the mitochondria is and what the level of the mitochondria is producing energy. And based on the different level, the mitochondria sends out numerous different factors, many of them, biochemical factors I will talk about. It also sends out electromagnetic signals, which different parts of the cell and other cells respond to as well, which are very important. It's a very underrated aspect of mitochondrial and even organism biology. But essentially, what's really important to get is that basically it was Doug Wallace's hypothesis and mine as well that this explains how a lot of diseases, and specifically one thing that I've seen clinically, is that you'll see some of these autoimmune diseases switch from one disease to another. For instance, you'll get a malar rash, and the person will get diagnosed with SLE, systemic lupus. And then a few months later, it'll switch phenotypes, and it'll get diagnosed as a Sjogren's disease, where they get the dry eyes and dry mouth. And so the question is, why does this happen? And it doesn't make any sense kind of when looking at it through our mainstream current view that we have. But what's interesting is that when you look at it from this perspective and you understand that most likely what happened is that there's simply a drop in mitochondrial energy production past a certain threshold, and when you drop the mitochondrial energy production past a certain threshold, you get a switch in disease phenotype. And it appears, I literally saw that case in clinic, and there's been, I've talked to many other doctors that practice, treating autoimmune diseases, and this is something that's, you see. I don't want to say it's common, but relatively. And so you see these things, and the question is why. And what the hypothesis is that this is the explanation that explains it. And so what's interesting too, and how this changes treatment, is that I've seen many cases and heard many stories like this of patients that have came to me. So doctors will spend months and years and thousands of dollars, hundreds of thousands of dollars, trying to pin the right diagnosis in some of these autoimmune conditions. When in reality, the foundational issue is the same, and it's mitochondrial energy production. And certainly, trying to get the right diagnosis, is important and plays a major role. But I think in situations like this, when you can see this disease-switching phenotype, if you have this perspective and you have this knowledge, then you understand that at a foundational level, the disease is functionally a mitochondrial energy production disease, and that you can treat that. And when you treat that, you'll be treating the disease no matter what phenotype it's currently in. Okay, so this is the second case study. This is also a case study that Dr. Doug Wallace did. All right, so this is a family that had a mutation in the mitochondrial DNA at the ND6 gene. It changes the proline at codon 25 to leucine. It's a very highly conserved proline at that spot, evolutionarily speaking. So this woman in her blood had 50% mutant ND6 gene, and so she had optic atrophy and cerebellar ataxia. Now what's interesting is her sister only had 5% mutant in her blood and was perfectly normal. And so this woman, and you can see the family tree right here, this woman had 3 different partners and had multiple children, and every one of her children had 100% mutant and they all died. So you can see, again, when you have a different degree of mutation in the mitochondrial DNA, which results in a different degree of energy production in the mitochondria, the lady that had 50% in her blood had optic atrophy and cerebellar ataxia. Her kids had 100% mutant at that spot, and they all died, and her sister only had 5% and was perfectly normal. So again, you can see when you drop the mitochondrial energy production past a certain point, you get a disease, you get a switch in phenotype. So what you see from the person can be different depending on once they hit that critical threshold and the mitochondrial energy production drops past a certain point in a certain tissue, you get a certain disease. And this is just 2 cases. Dr. Doug Wallace has had many cases like this, and they've been very interesting. Okay, so what they wanted to do was they wanted to prove that the mutation in the mitochondrial DNA that I just showed you and just talked about was in fact the cause of the conditions that they were seeing. in these people. So what they did was they made a mouse and they gave it the exact same defect and they studied it. So they took a culture of the mouse cells and they mutagenized them and selected for the mitochondrial DNA variants. So they gave it the same thing. They gave the ND6 mutation, proline 25 to leucine, and there was also a CO1 mutation, valine 421 to alanine. Those numbers are not important to memorize. But what's interesting is, so then they took the mouse, and again, so that lady had optic atrophy and cerebellar ataxia, and what they saw when they gave this mouse that same mitochondrial DNA mutation is they saw the exact same thing. So they have this really cool slide here where you can see the optic nerve of the mice. So this was the optic nerve of the mice with the mutations in the mitochondrial DNA. You can see the nerve fibers are extremely swollen, and that's basically from inflammation. And then the normal optic fibers are over here. So basically there's this selective loss of the thin fibers, sparing of thick fibers. There's demyelination of the nerves and increased reactive oxygen species, which results in inflammation and damage. So what's interesting though is that they actually found with this specific mitochondrial DNA mutation, there's about a 30% reduction in respiration. So there's a defect in a drop in the mitochondrial function. But what actually happened was, as they put on more stress for these mice to create more energy, they saw a drastic increase in reactive oxygen species. So basically, when you drop the mitochondrial energy production in this specific example, the mitochondria was basically able to adapt in a way and able to keep up with the necessary level of energy production for the tissue. But at with this mutation, the cost of it was that there was a drastically increased reactive oxygen species and inflammation in order to be able to keep up with it. So again, you can see that when you have mutations in the mitochondrial DNA, and those mutations result in drops past the normal energy level that's supposed to be in the tissue, there's basically consequences, and we get the disease phenotype when that happens. Okay, so that's been my hypothesis, and we went over a couple of cases. And so what are the mechanisms for this? So the mitochondria controls the energetics of the cell, obviously. It controls the reactive oxygen species, as we just showed, which is associated with aging, disease. It's also an important second messenger, which is an underappreciated value in cell biology. It controls the epigenetic expression. So like I said before, the mitochondrial energy level, depending on how healthy your mitochondria are functioning, they send out these biochemical intermediates from the Krebs cycle and different things that actually go and affect gene transcription and literally control what genes get transcribed and turned on and turned off. And so it's not appreciated how strongly the mitochondria actually affects and almost controls the nuclear genome to a large extent. And obviously there's many factors that are affecting gene transcription, but the mitochondria is a major controller of the nuclear genome. And it's interesting that many people think that the nuclear genome controls the cell, but in reality, something has to turn those genes on and off, and the mitochondria and the mitochondrial energy production, the health of the mitochondria is actually really what's controlling a large function of that. And what's interesting too is not only does the mitochondria control by sending out biochemical intermediates, but also sends out electromagnetic signals and specifically biophotons. I'm going to talk about that more in a second. Okay, so the mitochondria also controls the integrative stress response. So it has a huge controlling factor in the immune response. It also controls adaptation. So if you want to increase your cardiorespiratory fitness and your VO2 max, the mitochondria is basically what is adapting to that and plays a huge role in being able to increase your cardiorespiratory fitness and your adaptations. And like I said, it has enormous control of the innate and adaptive immunity. And Dr. Doug Wallace has this really powerful quote where he said that the mitochondria literally regulate all aspects of human biology that are important to adaptation. And so adaptation is an extremely important concept for organisms of all levels of species, and of course for humans as well. And what's important is that, again, when we want to get stronger, or if we want to increase our cardiorespiratory fitness, which are both really important aspects of human health, we need the mitochondria, and the mitochondrial health is basically what's being affected and what is allowing for and being affected and control the adaptations. Okay, what is also interesting is that The mitochondria is literally an environmental sensor, and it's sensing both biochemical signals and electromagnetic signals in the environment, and then it's sending out signals, both biochemical and electromagnetic, to the rest of the cell and the other cells based on that information. So the mitochondria is really the integrator of the environmental signals that it's getting, and then it sends out its own signals to then turn on and turn off genes and to regulate other organelles and other parts of the cell and even other cells, in fact. And so it's really not appreciated, like I was saying before, how strong the mitochondria is actually controlling and running many aspects of the cell, and in turn us. Okay, so the mitochondria controls the epigenome. So like I was saying earlier, the mitochondria integrates the nuclear mitochondrial gene expression. And essentially what happens is if you perturb the system and you perturb the mitochondria and you basically stress them out, you send out these biochemical molecules that then regulate the epigenome. So again, it regulates the nuclear genome. It regulates nuclear gene expression to hopefully maintain homeostasis and normal health. But if you cannot, maintain that, you get disease. And so what's also not appreciated is that the mitochondria actually controls the release of almost all inflammatory mediators, including many of the cytokines. Okay, so mitochondria plays an enormous role in immune system health. Now, what's interesting and I think worth mentioning is that, so I talked about how the mitochondria sends out many biochemical signals. That's well known in just mainstream biology and biochemistry. But what's not really appreciated as much and is not as well known but has been established for many decades in the literature very conclusively is that the mitochondria actually emits light, and it emits biophotons. And there's an amazing book called Light and Shaping Life, written by Roland Van Wyck. He's a scientist that's been studying biophysics for a long time, and he's got many papers published, and there's many other scientists that have published on this area too. But essentially, it's been experimentally proven over 50 years that the mitochondria emits biophotons, as it does metabolism and oxidative phosphorylation, which is the process that it makes energy by. And at first, it was uncertain if this was of any clinical significance when they were discovering this many decades ago. Then it was discovered that it does indeed have biological effects. And now at the cutting edge of biophoton research, it appears that it's playing a major role in regulating all aspects of human biology and biochemistry. And so basically, not only is the biochemical mediators that the mitochondria releases important in regulating cell function, but the biophotons and the actual light, the literal photon signals that the mitochondria is emitting, we now know is playing a major role in regulating many aspects of cell function and even organ and systemic function. Okay, so I want to talk about mitochondria specifically with respect to neuropsychiatric disorders now. Okay, and we're actually going to go back to that second case study. So this was the same, this is actually the same mutation. So this individual had that mutation that changed the proline to leucine at codon 25 and ND6 nucleotide position at 14600. Not important to memorize. But the woman had 50% mutant in her blood, so if you'll remember, she had that optic atrophy and cerebellar ataxia, and then the sister had 5% in the blood. She was perfectly fine. Okay, so they created the mouse germline that I talked about earlier with the ND6 mutation that changed the proline 25 to leucine. So this resulted in a 60% reduction in the respiratory complex 1, and then the reduction in complex 1 causes buildup of NADH. It reduces the electron transport chain. It creates a lot of electrons that make ROS. And like I talked about, I showed you the slide of the optic fibers. They had a lot of inflammation and reactive oxygen species release because of the defect in the mitochondrial DNA and the resulting loss of mitochondrial energy production. And so in order to basically make up for that, there's a drastically increased reactive oxygen species and inflammatory outburst. Okay. All right. So they compared the mitochondrial DNA mutations. So there was a guy named Dr. Ryan Morrow, did some really cool research. He isolated the mitochondria from the cerebral cortex of these mice. So again, we're comparing the ND6 mutation, then they compare that to a different mutation, ND5 mutation. So what they found interesting was that there's a direct proportionality between behavior and the mitochondrial cortical defect. And so Dr. Doug Wallace, based on the research, he said, based on the evidence, we believe that all neuropsychiatric disorders are about mitochondrial dysfunction. And because essentially what they did was they put these mice through these different mazes. They did, if you can see all the studies here, put them through mazes. They tested them out with different objects and different social interactions and studied the way they behaved with these different mutations. And again, they found a direct proportionality between the behavior of the mice and the actual mitochondrial DNA mutation and the level that the mitochondrial energy production dropped to, which gave them these conditions. So they tested for depression in these mice with this ND6 point mutation, and they found that they can literally give mice depression by creating a specific point mutation in their mitochondrial DNA at ND6, which was extremely fascinating. The mice were also tested for OCD. They put it into a cage where there was a novel object and a novel mouse, and most normal mice, they tend to visit with the novel mouse, but these mice looked at the, they tended to look less at the novel mouse, and they looked more at the object. They were hypercompulsive and had OCD, and they also had autism, which explains the symptoms here. And so what Dr. Doug Wallace said was that a single point mutation in the mitochondrial DNA resulting in a 10 to 20% reduction in mitochondrial respiration, which means mitochondrial energy level drops by 10 to 20%. They found that it generates all the neuropsychiatric disorders, and they've seen that both clinically in the case study 2 that we went over earlier, and then they basically tried to prove causality in these mice, and they found that it basically was correct, that it appears to be causal, that when you get this ND6 mutation and the energy level in the mitochondria drops below a certain level, in the brain specifically, then you get these conditions. You get these phenotypes. Okay, so I want to talk more about how the mitochondria affects the immune system and controls a lot of immune function. So when you see these cytokine storms, Epstein-Barr virus, Lyme disease, so there's many of these viral illnesses that, especially in naturopathic medicine, functional medicine, we see and deal with a lot. And what's been interesting to me for a long time is that, so I looked up the statistics on Epstein-Barr virus, for example, and over 99% of the population has come into contact with Epstein-Barr virus. And some people, some practitioners will demonize Epstein-Barr virus and these different other viruses, and they'll say that the virus is the problem. But what's interesting is that a very small percentage of people end up having an actual problem with these viral diseases. And so the question is, why does over 99%, almost everybody, get exposed to, for say example, Epstein-Barr virus, but only a very small percent of people, basically get affected by it significantly and go into this chronic viral syndrome where they have this chronic inflammation, chronic fatigue, brain fog. Sometimes they have anxiety and different things. And so the question is, why are most people okay and some people, basically struggle with it and it significantly affects their lives and gives them these chronic fatigue and things like this? And so based on Dr. Doug Wallace's research and others, what they're finding is that people who basically have a very robust system and specifically have a very robust mitochondrial function where their mitochondria are very thermodynamically efficient and are producing energy at a high level, when they come into contact with many of these viruses, they basically have no problems. And what's interesting is that they find that when people who have a decreased mitochondrial energy level, decreased thermodynamic efficiency at the level of mitochondrial energy production, and also just a decreased basically health of the mitochondria, these people are much more susceptible to, when they come into contact with a virus, they're much more susceptible to basically being susceptible to it and have the chronic fatigue and having all the symptoms that affect these people's lives very significantly, and are basically get sick from it. And so it's my personal hypothesis that this is what's happening in many of the cases of Epstein-Barr virus, Hep C, etc., and these other chronic viral illnesses where the small percentage of the population is getting affected by it, where most people are perfectly fine. And it's my hypothesis that this is why that's occurring. Okay, so what's interesting is we've known in the epidemiology literature for a very long time that certain people, certain ethnic groups, when they're in certain regions, they're more susceptible or less susceptible to many chronic illnesses, whether it's diabetes, autoimmune disease, etc., than other ethnic groups. And what's interesting, and what Dr. Doug Wallace has done a lot of research on, is that what they're finding is that when people are way outside of their basically where their ancestors were from and where they adapted to live. For example, if your entire heritage, for example, had come from sub-Saharan Africa, and now you're living, say in northern Canada, you're barely getting any sunlight, and basically you're just completely in a different environment than what your mitochondrial lineage had adapted to. We find that these people in studies do much poorer with chronic diseases than people who are living in a much more an environment that is much more suitable to their ancestral mitochondrial lineage. So people who are living in a place that their mitochondria is not adapted to, in the epidemiological literature, fare much worse. They get much more diabetes, much more autoimmune disease, cancer, and all these things, because basically what they found is that the mitochondria actually adapts to where people are living. So, people who are living in the sub-Saharan Africa, for example, have much different mitochondria than people who are living in northern Europe. So people that were living in sub-Saharan Africa, the mitochondria, the cytochrome proteins within the mitochondria are much more packed together. And what this does is it gives much more efficient, thermodynamically efficient energy production in the mitochondria, and it generates very little heat, which is very useful because there's not an abundance of food usually, and it's already very hot, so you don't want to be producing much heat. But people in northern Europe, so what they did is they uncoupled their mitochondria, so their mitochondria adapted the cytochrome proteins that make energy in the mitochondria spread out, and that allowed for increased heat production in the mitochondria, and it also lowered energy production, so we were less efficient at burning calories, but we were creating heat that allowed us to survive the very cold winters that occur in northern Europe, for example. So your mitochondria are very specifically adapted in that way and several other ways as well. And so when you're outside of the environment that you were essentially adapted through your ancestral lineage to be, we find that these people have much more chronic diseases than people who don't. And additionally, if you were to say run into a virus when you're in a place that you were not adapted to versus a place that you were adapted to, people who are living far outside of their ancestral mitochondrial lineage where, say they weren't adapted to it all, these people tend to have a higher susceptibility to getting more sick compared to people who are living in a place that's much more suited and where their mitochondria would have adapted over many generations based on their ancestral lineage. Okay, so like I was talking about earlier, we think that's why some people are more or less susceptible to different chronic viral illnesses based on basically the environment that their mitochondria have been in their ancestral lineage. And that's what I was talking about with the coupled versus uncoupled mitochondria, where the cytochrome proteins are either tightly together if all your ancestors were in sub-Saharan Africa, or the cytochrome proteins are more spread out and they uncouple for people that were in have a northern European descent, for example. Okay, so and as I've talked about before, the mitochondria controls much of the immune function, both the innate and adaptive immunity. However, what it also controls, and this was just such a fascinating point, so it just so happens that the inflammatory T cells in the immune system are glycolytic. So what that means is they don't really rely on mitochondria to generate energy, and they run much more on glycolysis, which is the burning of glucose in the cytoplasm. But the cells that put the brakes on the immune system and basically dampen down the inflammation and suppress inflammation, basically the population of T suppressor cells, they are loaded with mitochondria, and they run off of mitochondria making energy, which is called oxidative phosphorylation. And so why is this significant? Well, basically, people who get the cytokine storms, for example, when they have when you have systemic mitochondrial dysfunction, it doesn't really affect the inflammatory T cells. So the immune cells that actually make inflammation in the body, they can run rampant and make tons of inflammation and reactive oxygen species systemically throughout the body because they're not affected by mitochondrial dysfunction because they don't run off of mitochondrial dysfunction. However, when you have that decrease in mitochondrial energy production systemically, it affects the cells that actually decrease the inflammation, these T suppressor cells. So these cells are affected. So you get this situation where when you have systemic mitochondrial dysfunction, and especially that's affecting these T cells, that the cells that create the inflammation are not affected, and so they can run rampant and do their job, whereas the cells that are supposed to turn off the inflammation are very highly affected, and they can't do their job because they have a decreased mitochondrial energy production, and they essentially can't do their job. And so in the research, they actually think that this is the reason why these people get these cytokine storms, because they have systemic mitochondrial dysfunction, and their T suppressor cells aren't working because they have mitochondrial dysfunction, and so the energy level just isn't adequate enough to be able to properly put on this braking response and put the brakes on the immune response, essentially. Very interesting research. Okay, so speaking of interesting research, so this is some of the most fascinating thing that I've come across, and that's cancer and Dr. Thomas Seyfried's work, especially in correlation with Dr. Doug Wallace's work. Combining those two and the knowledge that they have shared has been very fascinating. So basically, we've kind of touched on how basically the hypothesis is that essentially every major chronic disease is a tissue-specific, energy level-specific mitochondrial disease, every major chronic disease. And so Dr. Thomas Seyfried has dedicated his life to cancer research, and he has essentially found that so the initiation of a normal cell to turn cancerous is impossible without defective oxidative phosphorylation, which again is how the mitochondria makes energy. So if you don't have defective mitochondrial energy production, the cell cannot turn cancerous, and we're going to talk about the studies that basically prove this in a second. And so the hypothesis is that cancer is also fundamentally a mitochondrial metabolic disease, and not a disease of nuclear genes, which is precisely what the current paradigm is. Okay, so in every study Dr. Seyfried's ever performed, there had to be defective oxidative phosphorylation and electron transport chain in the mitochondria, which is how the mitochondria produces energy, for the cell to turn from a normal cell to a cancerous one. There's not a single universal nuclear gene mutation present in all cancer cells, not even p53. I think that's an important point to highlight. So they've looked for nuclear genes. They've basically been looking at all these nuclear genes and trying to find if there's a single universal gene that is mutated in all cancers. And there's not a single nuclear gene that they have found that is mutated across all cancers, not one. However, what is present is that in every single cancer cell that turns from a normal cell to a cancerous one, there had to be defective oxidative phosphorylation and mitochondrial energy production. And this is basically Dr. Thomas Seyfried's work and what he's shown. And all right, so this is one of the more interesting things I've ever come across. So I call it basically the most interesting study I've ever seen done. So this was a genius set of experiments where basically the researchers took a normal cell, and they put healthy mitochondria into the cell, but they put cancerous nuclear genes into the cell. So they take a cell, they put healthy mitochondria into the cell, but cancerous nuclear genes, and they found out that the cell did not turn cancerous. So even when you put cancerous nuclear genes into the cell, as long as the cell has healthy mitochondria, the cell did not turn cancerous. Then they inverted the study. So they took a healthy cell and they kept the healthy nuclear genes of the study, but they took out the normal mitochondria and they put diseased mitochondria into the cell, and then the cell actually turned cancerous. So even when you have the nucleus of a normal cell, when you have diseased mitochondria, again, below that certain energy level, you actually get cancer in that cell. So what this basically almost conclusively showed was that cancer is basically a disease of mitochondrial dysfunction, because even if the nuclear genes are healthy, if the mitochondria are diseased, you will still get cancer. But even if you have a quote unquote like cancerous nuclear genome, as long as the mitochondria are healthy, it literally suppresses the formation of cancer, and you cannot get cancer if you have healthy energy production in the mitochondria. So this is some of the most fascinating research I've ever come across. And this could be an entire lecture in and of itself, but I hope I did an okay job of basically giving the gist of what this research was. Extremely fascinating research. And so, long story short, Dr. Thomas Seyfried he's a PhD. He's not a clinician, but he has gotten to do consulting on a number of cases. And long story short, they've had really great success when they're able to use his methods and look through a mitochondrial lens. They've had some really cool cases that you can look up on YouTube where he's talked about how they've actually gotten tumors to shrink when they look at things through a mitochondrial lens and treat it with mitochondrial biology. And again, things like surgery do play a role, so you can actually get the tumor to shrink using mitochondrial medicine, basically, and an understanding of the actual metabolism of cancer cells. And then you can use like surgery once the tumor has shrunk to basically go get the tumor out if that's clinically indicated according to the surgeon and the team that's treating the patient. But again, it's just really important because again, when you're basically increasing the overall health of the organism by mitochondrial energy production, you're also drastically reducing the risk that the cancer is ever going to come back. And I think when you view cancer through a mitochondrial lens, it makes so much more sense why so many cancers return, because if you're not fixing the root problem, which is the fact that the mitochondria are basically damaged systemically, then it would make a lot of sense that the cancer will eventually return in a few years. And so I think there's major implications to this research, and I think we're not fully utilizing the findings here to change our thinking in clinical medicine with respect to cancer, but I think we really should be. Okay. So moving on from cancer, I want to talk a little bit more about how the mitochondria specifically adapts to different places. So what's interesting is they actually used mitochondrial biology sequencing. So they actually sequenced the DNA of mitochondria, and that's actually how they figured out how humans migrated across the world. So just kind of a very fascinating fact, and kind of shows the importance of the mitochondria again. But what's interesting, and the reason they were able to do this is because the mitochondria actually adapts very specifically according to the area that you're located. And the reason basically that it can is because, so unlike the nuclear genome, so the mitochondria is actually an ancient bacteria. There's called the theory of endosymbiosis, where basically the mitochondria used to be a bacteria that hopped inside or was phagocytized by an ancient archaea. And so the mitochondria is actually an ancient bacteria. And what we know about bacteria is that bacteria can adapt, and in fact, they do adapt. So we have this very adaptive bacteria that's basically now the engine that's running our cell and our health. But it's important to note that the mitochondria adapts very specifically to different areas on the planet. And so if your entire ancestral lineage came from equatorial Africa, for example, and now you're living in northern Canada, we know epidemiologically that you're going to be at a significantly increased risk for all of the chronic diseases. And so this is something that clinicians and doctors are not talking with patients about, but it needs to be brought up and discussed because it's a significant factor. Okay, so this is clinically relevant, basically, like I said, because the mitochondria adapts to specific areas, and if you're living in an area that your mitochondria are not adapted for, then you're at a drastically increased risk of disease. And specifically, I want to talk about one way that the mitochondria adapts. So the mitochondria and the cytochrome proteins in the mitochondria specifically can uncouple or recouple. And so the spacing of these proteins on the inner mitochondrial membrane in the mitochondria, when we were at equatorial Africa, so when humans basically, long story short, they think that humans originated from basically around sub-Saharan Africa region. And so when we were at that area of the world, our mitochondria were extremely tightly coupled. So the proteins in the inner mitochondrial membrane that are the ones that make the energy were extremely close together. And what you get with that is very little heat production and very tight thermodynamic energy efficiency. So you're producing energy very efficiently. And the benefits of that are obviously that, when you're in Africa, you don't want to be making excess heat because it's already hot, and you want to be producing energy as much as efficiently as possible because there's not a ton of calories in the environment. However, as we moved and migrated across the world as humans, as we moved into, say, Northern Europe, for example, or even just Europe in generally, the calories were more abundant, but it was a lot colder, especially during the winter. So what actually happened is our mitochondria adapted, and these cytochrome proteins actually spaced out more. So even though we basically lost thermodynamic efficiency of our mitochondria, so they weren't producing energy as efficiently. However, they were producing heat, which kept us alive during cold winters. So that's one of the ways that the mitochondria actually adapts. And that's a very important mechanism to understand. Okay. So basically from the information of your ancestral lineage, you can determine how coupled versus uncoupled a person's mitochondria is, and that's a significant factor to a person's health and thermodynamic efficiency, basically. And people, additionally, so it explains a lot of metabolism and how, so people that basically have high metabolism, these people are very likely to be uncoupled. So their mitochondria cytochrome proteins are spaced out, so they don't convert energy as efficiently. So these are people that they can eat a ton and they don't gain as much weight because they're not converting the calories that they eat as efficiently as somebody who has very coupled mitochondria. So it's a major factor in the obesity epidemic that this country is facing, and it's a very important factor for many other health issues as well. So this environmental mismatch can be a big risk factor, which is kind of the main point I wanted to talk about there. Okay. So to wrap this up, I kind of want to take a step back and kind of look at modern medicine and how we got to where we are today by looking at things through a nuclear genome lens and such a reductionistic view, and how I think we should move forward. So a lot of the reason that medicine is set up the way it's set up is because what actually happened in the history of medicine is we found out, and our knowledge of anatomy progressed much quicker than it did our knowledge of biochemistry. So basically anatomy was being kind of discovered and unfolded around the 1500s. This guy named Vesalius basically is considered the father of modern anatomy. So we were basically discovering anatomy almost 500 years ago, and even around the 17 and 1800s, we were really starting to uncover a lot of the major relevant anatomy. Whereas our knowledge of biochemistry really did not get going until Albert St. George and Hans Krebs kind of discovered the Krebs cycle and got that area going around the 1930s and 40s. And so you can see that our knowledge of biochemistry, and then especially of biophysics, really lagged behind our understanding of anatomy. And so the way that we originally came up with our medical system was that we understood the anatomy first, and so we decided that if you have a disease in a specific or symptoms in a specific area of the body, you would go see that specialty. So we specialized our medicine based on anatomy. And so, for example, if you have a headache, your primary care doctor might refer you to go see a neurologist because basically they're saying there's something wrong with your head. But what Dr. Doug Wallace has found and said is that just because you have basically an issue in your head and your brain, that doesn't mean that the problem's there. It's just as likely that you have a systemic mitochondrial dysfunction for which the head is much more sensitive to than the rest of the body. So basically, again, what you have happening is that there's a systemic drop in mitochondrial energy production and energy efficiency. And the brain, as most people know, is about 2% of the body weight, but it takes up about 20% of the blood flow. So the brain is extremely sensitive to drops in mitochondrial energy production. And so when the brain can't produce enough energy, you get these headaches. Again, as Dr. Doug Wallace has shown and theorized, basically all the major chronic diseases are drops in energy production below a certain threshold. And there's, clinically, we know that there's many causes of headaches, but basically, long story short, the hypothesis that I presented and that Dr. Doug Wallace and others have shown is that it's much more of a mitochondrial systemic problem where the actual mitochondria and the systemic health of the organism through energy metabolism is basically what's responsible for whether you're healthy or whether you're sick. And so we have, in modern medicine, we have such a reductionistic view where we're taking such a myopic, narrow view at the problem and only looking in the area of the problem, and we're all basically only looking, at the anatomy of the area where the problem's at. So looking at your head, when in reality, we need to be looking at things like diet, like exercise, like light exposure, like sleep, like environmental factors, and taking a much more holistic, systemic view instead of having a very narrow view onto just a certain area of the body and the anatomy associated with that area. And again, so specialists in medicine definitely have their place, and they definitely serve a role in society and in medicine, and we're thankful to have specialists. But I think medicine in general needs to firstly take a much more holistic view of health, disease, and the organism, as opposed to the extremely myopic, reductionistic view that we currently have. And additionally, I think it needs to take a mitochondrial view, basically, of all the of health and disease, as opposed to an anatomical view, which we currently have. Okay, and then the second paradigm that I want to just kind of talk about is that, so this scientist named Mendel, basically, he was the one that basically came up with the pea plants and the crossing of genes, and basically kind of was the first one to kind of start discovering how genes worked. And then Watson and Crick basically kind of cracked the genetic code, and we started the Human Genome Sequencing Project. But basically, long story short, they thought that we would solve like every problem when we cracked the genome, but clearly we have not done that. And so basically, what I'm proposing, what Dr. Doug Wallace and others have proposed, is that the mitochondrial and the energy production, the level of energy in the cell and in the organism is much more important than the nuclear gene. And I hope I did a good job of showing why, because the mitochondria is actually what controls much of the epigenetic expression, much of the actual expression of the genes, and not the other way around. And again, I think the cancer study that I talked about, where they replaced the mitochondria and then they replaced the nuclear genes, I hope that kind of showed the power of how the mitochondria actually controls the expression of the cell in such a powerful way. And so lastly, this I just want to wrap this up with how this changes treatment and clinical thinking, and hopefully how it changes medicine. So obviously this changes the treatment paradigm for many diseases. So when you're looking at a disease through a mitochondrial biology lens instead of a nuclear genome lens, I think the treatment plan looks completely different for almost all of the major chronic diseases, including cancer, neuropsychiatric conditions, things like this. So basically the treatment plan looks completely different when you're looking at things through a mitochondrial lens instead of a nuclear genome lens. And so I think medicine really needs to change in that respect. Another important thing to look at is when you understand this, so when you understand that how important mitochondria are, and you understand that all of your mitochondria is inherited from your mother, then I think you start to look at the family history completely differently and look at the diseases and health on the mother's side a lot more closely than people were in the past. And so I think that's an important concept that I try to get to my colleagues. And then additionally, one of the other things so mitochondria are bacteria, and because bacteria can rapidly adapt far more so than our nuclear genes can, this really changes the paradigm of what's possible. And so I've been fortunate enough to work with some patients. I've had patients who had specifically, I had a patient who had Hashimoto's disease. We diagnosed it with clinically with symptoms, and then also the anti-TPO antibodies were 336. The reference range is below 9. And then about 2.5 months later, we retested, and the antibodies were back, basically non-detectable, and all of the girl's symptoms were completely gone. And so that's just one case of basically what is possible when you look at things through a mitochondrial lens instead of looking at things through a nuclear genome lens, where you think that person was destined to get that disease, when in reality that's not the case. And when you basically treat the mitochondria using mitochondrial medicine, you can actually get very incredible results using this type of thinking and this type of medicine when you understand the actual disease at its root and, in my opinion, what should be done to treat it. Okay, and then the last point is that mitochondria are responsive not just to biochemical things, but to electromagnetic signals from the environment. This is something that we are really doing a poor job of understanding as a medical community. But not only are the mitochondria sensitive to biochemical pollutants such as pesticides and herbicides, but Dr. Doug Wallace and other researchers have clearly shown we've known this for decades, that the mitochondria is actually sensitive to electromagnetic signals across a very wide range of spectrums. And so it responds. So sunlight actually affects mitochondria basically in a beneficial way as long as it's appropriately dosed. But then additionally, so non-native EMFs, which are very ubiquitous in society, also mostly negatively, are impacting mitochondrial health. And so I don't think many people at all are really considering the benefits of appropriately dosed sunlight exposure and the negative effects of things like non-native EMFs, which are very common in society. But when you understand that mitochondria are sensitive not only to biochemical inputs from the environment, but also electromagnetic inputs from the environment, then you can understand how this actually is significantly affecting mitochondrial function. And I think then more clinicians would talk about appropriately dosed sunlight exposure, as well as avoiding non-native EMFs as best they can. And so that is the end of my presentation. Thank you for watching.